blood cells, but 2% plasma, but we ignore this percentage, the trapped plasma. Now we'll start talking about the production of the blood cells, mainly actually the red blood cells. From fetal life till adults and old age. You see, during the fetal life, before birth, blood cells mainly produced in the or by the liver. From the second month till birth. Little produced by the spleen. In the beginning of the 2 months, blood cells produced by yolk sac. The bone marrow begins to produce blood cells at the beginning of the 5th month. See? And continues till the end of life. Lymph nodes also along with the bone marrow, but usually after birth they don't produce. After birth, blood production occurs only in the bone marrow, usually. But sometimes in abnormal cases, liver and spleen, after birth, they produce blood cells, but in abnormal cases. Blood production is called erythropoiesis or hematopoiesis. Till the age of 18 to 20, all the marrow in all the bones produces blood cells. But after being adult, in males or females, blood production is confined in some bones, mainly in the vertebra, pelvis, sternum, ribs, femur, and tibia. Then you see, after being adult, the erythropoiesis occurs in specific bones, but the highest in vertebra, then pelvis, sternum, ribs, femur, and tibia. Regulation of erythropoiesis. Normally the number of the blood cells, whatever erythrocytes, white blood cells or platelets, they remain constant. They keep normal concentration in the blood. As you said, 5 millions of RBCs from 4 to 11,000 of white blood cells and platelets or thrombocytes from 150 to 400,000. This number is kept normal during the life. The change is very slight. It means that the production is equal to the destruction. The production of blood cells equal to the destruction of the blood cells. For example, the red blood cells, they function for about 120 days. Then they are destroyed. To replace this destroyed or damaged or these old blood cells or erythrocytes, they have to be replaced by new cells. This is the normal erythropoiesis. Production equal to the destruction or damaged blood cells. First factor regulates the erythropoiesis oxygen supply to the tissues. You can understand that oxygen supply to the tissue must be relatively constant. Low oxygen supply is called hypoxia. High oxygen supply is called hyperoxia. In hypoxia, to compensate the low oxygen supply, red blood cells count increases. In hyperoxia, red blood cells production decreases because there is high oxygen supply. Therefore, we can understand that when the baby is born, the blood cells, especially the red blood cells, count is high, but gradually decreases because of the normal oxygen supply from the environment. The oxygenation of the baby through the lungs. Another, for normalizing the red blood cells count, the replacement of fetal. You see, when oxygenation, tissue oxygenation decreases, whatever the type, the decrease in tissue oxygenation, either through low blood volume, or anemia, or low hemoglobin, or poor blood flow, or pulmonary disease, whatever. When tissue oxygenation decreases, there are cells in the kidneys. These cells, they are sensitive to the reduced tissue oxygenation. When the tissue oxygenation decreases, the cells in the kidney, especially cells, they produce hormone called erythropoietin. This erythropoietin affects the stem cells in the bone marrow to increase the red blood cells in the blood. This is how the reduced tissue oxygenation increases the red blood cells in the blood by increasing the production of hormone called erythropoietin by cells in the kidneys. Erythropoietin is a glycoprotein hormone. We can trace it in plasma or in the urine, stimulated by hypoxia and inhibited by hyperoxia. 90% of the erythropoietin hormone produced by the kidneys and 10% by the liver, and maybe by the spleen. Even the production of erythropoietin, either renal or extra-renal source. You see here the role of erythropoietin. And here, in this figure, shows the stages of the production of the red blood cells or mature red blood cells. The stages, or these stages you study in histology. You see, the last stage of the production of the red blood cells, the reticulocytes. Reticulocyte cells, they are present in the bone marrow, in the blood. The percentage of reticulocytes in the blood ranging from 1 to 2. When the percentage of the reticulocytes in the blood about 1 or 2, it indicates normal erythropoiesis. Below that indicates low production. Above that indicates high production of blood cells. The number of reticulocytes in the bone marrow is about equal to the number of the reticulocytes equal to the number of nucleated red blood cells in the marrow. But the reticulocytes in the peripheral blood are slightly less than that of the bone marrow. You see, hemoglobin synthesis occurs in all blood cells in the bone marrow, including the reticulocytes. But hemoglobin synthesis does not occur in the mature blood cells. And even in the reticulocyte in the peripheral blood, the hemoglobin synthesis does not occur. The second factor which regulates the erythropoiesis vitamins, mainly vitamin B12. Actually, all vitamins are needed for normal erythropoiesis, but vitamin B12 is essential for normal erythropoiesis. Vitamin B12 is called also extrinsic factor or maturation factor. Vitamin B12, we take it in the food. This vitamin B12, when is taken from the food, unites with the protein produced by the gastric mucosa, intrinsic factor. Vitamin B12 bound to intrinsic factor transported into the lower ileum. It means that vitamin B12 cannot be absorbed properly or affected by the enzyme if this intrinsic factor is not produced by cells in the stomach. Then from the lower ileum, vitamin B12 is absorbed, transported into the blood circulation, participate in the blood production, or stored in the liver. This is the absorption of vitamin B12 or extrinsic factor or maturation factor. Vitamin B12 also essential for the production of the neutrophils. The researchers noticed that in vitamin B12 also neutrophils number or count is reduced. The anemia which occurs because of the vitamin B12 is called pernicious anemia. الأنيميا الخبيثة or megaloblastic anemia. The reason for the reduced count of the red blood cells in pernicious anemia, the vitamin B12 is needed for the maturation of the red blood cells, for the DNA formation. DNA formation time is prolonged, but the hemoglobin synthesis is normal. When the time for the maturation of the RBCs is prolonged, the hemoglobin synthesis increases. Therefore, the red blood cells produced having MCV high, more than, let us say, 100 or sometimes 150 micron cubic, containing high hemoglobin concentration, but low count. Also oval in shape, the red blood cells produced, and fragile, they don't survive. They have short half-life. Also, in pernicious anemia or megaloblastic anemia, we notice that the cause of the pernicious anemia actually not the deficiency of vitamin B12 from the food uptake, but from other causes such as the deficiency of the intrinsic factor. Why? Because 2 to 3 milligram of vitamin B12 is sufficient for the human being for about 3 to 4 years. It means that the pernicious anemia, which occurs, is very, very rare because of the deficiency of vitamin B12 in the food, but from the other sources. The deficiency of vitamin B12 also affects the normal function of the myelin sheath. In vitamin B12 deficiency, the myelin sheath function also is affected. Now we'll see the cause of vitamin B12 deficiency. Cause of vitamin B12 deficiency, veganism, those they have just vegetables. Malabsorption, either gastric causes or intestinal causes. Gastric causes congenital lack of intrinsic factor or total or partial gastric tumour. Intestinal causes, for example, chronic tropical sprue, chronic diarrhea, or ileal resection, the site in which the vitamin B12 is absorbed. Then these are the causes of vitamin B12 deficiency. Also, from the vitamins, the folic acid, essential for the production of the red blood cells. Folic acid essential for the DNA formation, but it has nothing to do with the myelin sheath. Cells produced when the folic acid or folate deficiency almost similar to the cells produced because of the vitamin B12 deficiency. Cells are large, oval in shape, high MCV, short half-life, contain a lot of hemoglobin. Anemia produced is called megaloblastic anemia. Folic acid is absorbed through the jejunum, where there is enzyme there, carboxypeptidase, which is essential for the absorption of vitamin B12. Therefore, you can understand that the anemia which occurs because of the deficiency of vitamin B12 and folic acid is called megaloblastic anemia. But the anemia which occurs because of the folic acid is not called pernicious anemia. Megaloblastic anemia is applied to both because both cells occur because of the deficiency of vitamin B12 and folic acid are large cells containing a lot of hemoglobin. Therefore, they are called megaloblastic anemia. And these are the causes of folic acid deficiency or folate deficiency. Inadequate dietary intake, malabsorption, such as in celiac disease, jejunal resection because the folate absorption occurs there, and tropical sprue, tropical diarrhea. And 3 include requirements such as pregnancy, premature infants, chronic hemolytic anemia. And these are the causes of the folic acid or folate deficiency. Vitamin B12. These are definite effects of vitamin B12 or folate deficiency. Megaloblastic anemia in both cells having high MCV and a lot of hemoglobin. Macrocytic of epithelial cell services. In neuropathy, the effect on myelin sheath for vitamin B12 only, sterility, الحقن, and rarely reversible melanin skin pigmentation. Just to have an idea about the duration of erythropoiesis, normally from 3 to 6 days. Normally, the duration of the erythropoiesis ranging from 3 to 6 days. Now we come to as we said, all the vitamins are essential for normal erythropoiesis, but mainly, as we said, vitamin B12 and folic acid. And don't forget vitamin C, which is involved in every process in the body, vitamin C, even in the defense mechanisms.